Thank you for joining today's webinar. We'd like to introduce you AllView PAGE Buffer, an innovative running buffer which gives similar gradient separation profile using a single percentage page gels, and it has extremely fast electrophoresis run times. Before discussing AllView PAGE Buffer, we'd like to give you a brief background about Funakoshi Company. Our headquarters is based in Tokyo and has been dealing with life science market for over 50 years. Biodynamics Laboratory Incorporated is our subsidiary which developed and manufactures AllView PAGE Buffer, as well as pre-stained RNA markers, protein markers, ultraripid and other molecular biology and proteomic products. Polyacrylamide gel electrophoresis has been widely used for separating and characterizing protein sizes. PAGE can be classified into two major methods, SDS-PAGE or native PAGE. SDS-PAGE uses SDS detergent, and with a combination of SDS and reducing reagents, proteins are separated by electrophoresis. The proteins can be observed as bands with higher molecular weight proteins located upper side of the gel, while lower molecular weight proteins are located lower part of the gel. On the contrary, native PAGE does not use detergent, and whole protein subunits maintain three-dimensional structure. In this presentation, only SDS-PAGE will be discussed, not native PAGE. Protein samples vary in size from small molecular weight to large molecular weight. Polyacrylamide gels restrain larger proteins, while smaller proteins migrate faster in gel. In SDS-PAGE, several single percentage concentration of polyacrylamide gels may be used depending upon desired protein sample size. The most common single percentage acrylamide gels often used range from 6 to 15%. Gels with higher acrylamide concentrations are good for separating low molecular weight proteins, but not suitable for high molecular weight proteins. Gels with lower concentrations are good for separating high molecular weight proteins, but not suitable for low molecular weight proteins. This makes it difficult separating wide molecular weight range of proteins from one gel. Single percentage gels are not suitable for separating wide range of protein sizes. To overcome this problem, gradient gels are useful. A gradient gel consists of low acrylamide percentage at top of gel and high acrylamide percentage gel at bottom, allowing resolution of wide molecular weight protein range. However, purchasing gradient gels may be more expensive, and if preparing gradient gels in a lab is difficult and time-consuming. Biodynamics Laboratory has developed AllView PAGE Buffer. Simply replace electrophoresis running buffer with AllView PAGE Buffer. AllView PAGE Buffer gives us a wide range protein separation using 6% single percentage gel from high to low molecular weight, nearly equivalent to using a gradient gel. This is a comparison data between 6%, 15% gradient gels, and 6% with our AllView buffer. You can see here in the red box, using 6% gel with typical tris running buffer, high molecular weight proteins are clearly separated, but low molecular weight proteins are poorly separated. On the other hand, a 15% gel concentration, the resolution of low molecular weight protein is good, but resolution of high molecular weight protein is poor. In 4 to 20% gradient gel concentration, low to high proteins are clearly separated with good resolution for both high and low molecular weight proteins. On the far right side shows AllView PAGE Buffer with 6% homogeneous gel, gives similar protein separation resolution compared with gradient gel. While AllView PAGE Buffer separates proteins similar to gradient gels, another big advantage using AllView Buffer is running time. Running a gel normally requires 1 hour or longer. However, using our AllView PAGE Buffer, electrophoresis can be completed in 13 minutes at 250 volts or 18 minutes at 200 volts. Here's the product information for AllView PAGE Buffer. Catalog number D as in David, S as in Sam, 520. It comes in a 500 ml volume size with a stock concentration of 20x, which should be diluted down to 1x. You may store this at room temperature for 12 months. The recommended acrylamide concentration is 6% for resolving gel and 3% for stacking gel to separate protein range of 10 to 250 kilodaltons. If separating low molecular weight proteins, 10 to 30 kilodaltons, the recommended acrylamide concentration is 10% for resolving gel and 3% for stacking gel. AllView PAGE Buffer should only be used for SDS-PAGE. However, you should not use AllView PAGE Buffer with native page. Here are the electrophoresis running parameters. At 250 volts, you may run the gel for 13 to 15 minutes. At 200 volts, you may run the gel for 18 to 20 minutes. AllView PAGE Buffer is recommended for desired cost savings compared to purchasing gradient gels. There are several cost savings when using AllView running buffer. In general, pre-manufactured gradient gels cost more compared to hand-casted single percentage gels. There are labor cost savings hand pouring single percentage gels instead of hand pouring gradient gels. Finally, there are cost savings when running 2 single percentage gels with AllView buffer in electrophoresis apparatus compared to 2 gradient gels. Quicker gel results compared to general running buffer with hand poured single percentage gels. Using AllView running buffer allows fast electrophoresis run times as short as 13 minutes at 250 volts, giving quicker gel results. Using fewer single percentage gels to obtain full range protein separation. Some labs completely avoid using gradient gels, but will run 1 low percentage gel for high molecular weight proteins and 1 high percentage gel for low molecular weight proteins for a total of 2 gels to obtain the research data. Using AllView buffer, only a single 6% gel is required instead of using 2 separate gels. AllView buffer may not work in precast manufactured gels. Some ingredients in precast gels will interfere with AllView running buffer. AllView PAGE Buffer is intended for hand-casted gels, or check if our AllView PAGE Buffer is compatible with your precast gel before experiment. In summary, AllView PAGE Buffer is a special electrophoresis running buffer for SDS-PAGE. Separates wide range sizes from low to high molecular weight proteins using 6% single percentage gel. No need to buy expensive gradient gel or precast gels. Time savings, rapid electrophoresis run times as short as 13 minutes at 250 volts or 18 minutes at 200 volts. Faster electrophoresis run times gives experimental gel results sooner. In addition to AllView PAGE Buffer, we also have other related products from Biodynamics brand. We have pre-stained protein ladders stable for 4 degrees Celsius for 1 year, and a Kamasi Quick Blue staining solution. Thanks for listening to today's presentation. For additional information, such as pricing or to download the manual, you may visit Funikoshi's website in Japan. For United States and Canada, you may contact Diagnosing. For all other countries, please contact your local distributor.